name is Dr. Richard Nowak. I'm a neurologist, specifically an associate professor of neurology at the Yale School of Medicine, and I served as the global principal investigator for MINT, or Myasthenia Gravis Inebilizumab trial. So inebilizumab is unique in that it targets CD19 positive B cells. B cells are critically important in the production of antibodies, including pathogenic autoantibodies. As B cells mature, they have cell surface markers that are expressed, but some of those proteins are actually lost during the maturation process. What's unique about inebilizumab, as I noted before, is that it targets CD19 positive B cells. And these are the B cells that actually express CD19, but might lose the expression of, for instance, CD20. So we're more effectively able to directly target antibody secreting cells, specifically plasmablasts and certain long-lived plasma cells, which we believe are critical to pathogenic autoantibody production in autoimmune myasthenia gravis. In terms of inebilizumab's mechanism of action, it would target B cells that are antibody secreting cells. In MINT, we studied both acetylcholine receptor and muscle-specific kinase autoantibody generalized myasthenia gravis, which with those two combined comprise the majority of patients with generalized myasthenia gravis. So we would not necessarily be specific to only AChR positive or only MuSK antibody positive individuals with myasthenia. MINT was a phase 3 placebo-controlled international study that evaluated the safety and efficacy of inebilizumab in patients with autoimmune generalized myasthenia gravis. Specifically, it enrolled patients with symptomatic disease, either acetylcholine receptor or muscle-specific kinase antibody positive. Our initial primary endpoint was change in MG-ADL score at week 26. Our initial endpoint was met, showing that there was a significant difference in terms of the treated versus the placebo-controlled arm, demonstrating efficacy. Our initial safety analysis demonstrated safety and tolerability. What I am speaking of at the 2025 AAN is our 52-week readout, specifically looking at the acetylcholine receptor antibody subtype through week 52. For the acetylcholine receptor autoantibody subpopulation, the randomized control period was 52 weeks. We followed 190 patients in that particular arm of the study. And what we observed is that at week 52, there was a significant difference in the MG-ADL in the treated arm as compared to the placebo arm, demonstrating, again, efficacy, and also the QMG, which was one of our key secondary outcomes. We also looked at the percentage of individuals with a 3 or more point change at week 26 and also at week 52, and demonstrated a significantly reduced MG-ADL and QMG score as compared to placebo at both time points. In fact, the magnitude of reduction was greater at week 52 as compared to week 26. It appeared from our data that the patients in the treated arm were continuing to have greater improvements over time, looking at the percentage of patients with a 3 or more point improvement in their score, both on the ADL and also on the QMG. The placebo group seemed to be roughly about the same in terms of the magnitude of change. So that didn't really vary too much from week 26 to week 52, although, you know, the numbers and the differences there are not necessarily that great to really say anything more definitive. Anti-CD19 positive B-cell depletion therapy represents the potential first-in-class therapy for myasthenia gravis. Currently, we do not have an FDA-approved B-cell depleting therapy for patients with autoimmune myasthenia gravis. So I view this more so in buckets. So we certainly have 2 recent classes of medications, the FcRn antagonist, the C5 complement inhibitors. But this is an additional mechanism of action that could be utilized potentially for patients with antibody-positive generalized myasthenia gravis. Despite currently available therapies, there are individuals with inadequate disease control, and certainly those that have additional issues with the tolerability of certain medications. So having a strategy like this, I think, is important and might fill a currently unmet need. So that's what I would say. Also, I think it's also important to highlight the treatment schedule. So this is a medication that's given initially at day 1 and also then 2 weeks later. And then it's repeated again every 6 months as a single infusion. So the first year, individuals would get 3 infusions and then 2 infusions thereafter on an annual basis. So from a convenience or a treatment burden perspective, it has significant potential advantages, and patients might have a great interest in having something that's a bit more convenient and more frequent infusions or injection type medications.